My name is Eric Fried. I'm director of the HIV Dynamics and Replication Program at the NCI in Frederick. And it's a real pleasure to introduce the 2021 George Khoury Memorial Lecture. George was a graduate of Princeton University and Harvard Medical School. He came to the NIH in 1976 as head of the virus tumor biology section in the Laboratory of Molecular Virology, NCI. He became chief of that laboratory in 1980. In 1981, he received the Arthur S. Fleming Award for Outstanding Government Service. And in 1987, the year of his untimely death at age 43, he was elected member of the National Academy of Sciences. And here's a picture of George with two other very notable virologists, Peter Howley, who went on to Harvard, and Malcolm Martin, who is still here at the NIH and in fact was my own postdoctoral mentor. During George's brief career, he published over 140 papers deciphering mechanisms of eukaryotic transcription. And he had a very distinguished group of trainees and collaborators, some of whom are listed here and will be familiar to many in the audience. The lectures in this series have been given by a similarly eminent group of speakers, including 5 Nobel laureates. And I should point out that last year's speaker, or the last speaker we had in 2019, Julie Pfeiffer, trained as a postdoctoral fellow with today's speaker. So it's an honor to introduce today's speaker, Karla Kirkegaard. Karla did her undergraduate work at UC Berkeley, her PhD at Harvard, and then she went on to do postdoctoral work at MIT with David Baltimore. In 1986, she took an assistant professor position at University of Colorado at Boulder, received tenure there, and then in 1996, moved her laboratory to Stanford University School of Medicine. Between 2001 and 2005, she was professor and associate chair in the Department of Microbiology and Immunology at Stanford. Between 2005 and 2010 was professor and chair of that department. And since 2014, it's been the Violetta L. Horton research professor in the Department of Genetics, the Department of Microbiology and Immunology at Stanford. Karla has received a large number of honors and awards, far too many to list here, just listing several. She's an elected fellow of AAAS, member of the American Academy of Arts and Sciences, elected fellow of the American Academy of Microbiology, and in 2019 was elected to the National Academy of Sciences. Karla's research is currently focused on several high impact areas, including the identification of dominant drug targets for interfering with RNA virus replication, the role of nest long non-coding RNA in controlling microbial challenge, and the link between autophagy and pathogen infection. So without any further delay, it's a pleasure to introduce the 2021 Khoury Lecture given by Dr. Karla Kirkegaard, Quasi-species Suppression of Viral Drug Resistance. Karla, it's all yours.
Thank you so much, Eric, for that kind introduction. And also, I'm really enjoying being invited here. Last year, I was invited and we thought we'd put it off till this year so we could do it in person. And, you know, what can I say? We're all waiting for that to happen again. Okay, so I'm going to talk about, a topic in my lab that we've been working on for a while that I really want to focus on today, which is to the not so modest goal of rethinking antiviral drug targeting. And I'm really interested in feedback and collaboration from people who are also interested in this topic. If I were to summarize what I want to tell you today, I'm going to talk about positive-strand RNA viruses, which are the most plentiful class and the most rapidly evolving class of viruses. I want to emphasize and be grateful for this, the vaccine against COVID-19 virus, and also to emphasize that vaccines have in general been more useful in controlling RNA virus infection than antivirals. I'm leaving out HIV, which is a retrovirus, of course, and so we're talking about positive-strand RNA viruses here and negative-strand to some extent, which have, because of their iterative replicative cycle and the high error rate, believe it or not, even more variability than HIV. Therefore, you look at this antiviral list and it's pretty sad. The last several years has given rise to combination therapy for hepatitis C virus, and that's a great way to combat drug resistance. And remdesivir, which is, for COVID-19, whose utility has yet to be really well demonstrated, But you see the dearth of these. And why is that? The answer is mutation, mutation, mutation. And so that's what I'm going to address and ways to deal with that. Okay, I'm supposed to talk about the non-approved drugs and compounds mentioned, since there were all those dashes and there are very few antiviral compounds. Everything that I mentioned is pretty much non-approved, and the list is here and you'll see it again. All right, so we're going to talk about positive-strand RNA viruses. My lab has worked on poliovirus for a long time as a really great model system for all RNA viruses. We believe, all positive-strand RNA viruses, we believe. I'll also mention dengue and some of the work we've done with dengue in suppressing drug resistance. And of course, we're all thinking about SARS-CoV-2 and the lessons that we've learned through basic science over the years of controlling such outbreaks in the future. And, you know, vaccines are wonderful, and I'll actually refer to one of the reasons they're so wonderful, but I think we can all agree that antivirals would be helpful as well. Okay, all positive-strand RNA viruses, by definition, make one strand of RNA, pretty much, and that in the conventional dogma goes in alone when you infect with one individual viral particle. Okay, so this RNA is translatable. That's also the definition of positive strand. And what happens is a lot of spectacular cell biology once it starts making its proteins. Here, for example, is an image of a poliovirus infected cell. You can see the massive proliferation of vesicles. All positive strand RNA viruses of mammalian of eukaryotes replicate on the surface of membranes. And that's some great cell biology that we'll talk about some other time. What's relevant here are the sources of variation. So here's poliovirus again. And this single-stranded RNA encodes a polyprotein that's made all in one piece. That's a ribosome right there. And a lot of, I think all positive strand RNA viruses use a polyprotein strategy for at least some of their genome. So that's really interesting. So the first thing this positive strand RNA does is make this polyprotein. It gets cut up by various virally encoded proteases that are colorfully named things like 2A and 3C. We'll talk about them later. The first thing they have to do is cut themselves out of the polyprotein and their precursors, and then go on to be bona fide transacting proteases, and we'll return to that. Okay, you need proteins in order to do negative strand synthesis. You notice that these genomes are starting, these antigenomes are starting to be different colors. That's because they're accumulating mutations at a certain frequency that we'll also talk about in a second. Positive strand synthesis, we start to see more colors illustrated in the quasi-species that's being demonstrated, that's being generated as mutations accumulate. Okay, so before we get into that, I want to talk about what we mean and what people mean by when they talk about mutation frequencies and rates. So there's molecular misincorporation. That is, you have a template. We know that there should be a G and a primer, and we know that a G should be incorporated there, but no, what's the frequency of incorporating an A? And what we know on average from positive strand RNA viruses, this misincorporation frequency, not a rate, a frequency, is about 1 × 10⁻⁵, possibly lower for coronaviruses, which is a good thing. Okay, then there's a cellular mutation accumulation, which has to do with many cycles of this replication strategy, as you just saw. That's again a frequency, mutations per nucleotides per cycle. That averages 1 to 3 × 10⁻⁴ for genomes like poliovirus and dengue virus. Jury's a little out on coronavirus. But this is enough so that a 7,000 to 10,000 nucleotide genome like that of poliovirus or dengue virus, you know, has a few mutations per, on average, maybe 1 or 2 mutations per genome, which is of course going to be a Poisson. Okay, what we're talking about in terms of, you know, the evolution of viruses is given in a rate. There are rates such as mutations per nucleotide per year in an individual infected person for chronic infections and during transmission. How much does the virus change during transmission? And that, of course, depends on selective pressures as well as the intrinsic mutation frequency. So I just wanted to explain that because people use these different terms sometimes interchangeably, and it becomes very confusing. Okay, so I guess one of the main things I want to say and emphasize to understand this talk is that molecular diversity, because of the error of amplification of RNA, occurs within cells during RNA replication. This is not happening in mathematical space. This is often modeled. It's occurring, so if this one here, where the parent molecule, negative strands are made, then positive strands are made. And what you see is, genomes that are diverse within a cell and related by this quasi-species generation, cumulative mutation accumulation. So the question is, what happens now? What if this little pink genome here is, we're gonna have selective pressures operate on here. What if that little pink genome has a fitness advantage? Is it going to just grow out of this population? Well, that's what the mathematical models usually say, that this will grow out as a function of its fitness advantage and its frequency. But of course, that makes the assumption, which is sort of incorrect, that this viral genome, which has increased fitness, isn't helping its family, and more importantly, that its family isn't hurting it. And that's what we want to do. We want to figure out how to do what we call dominant drug targeting, in which this family into which this fit genome is born, for example, the drug-resistant genome, is interfered with by its family. And I'll get to that now. Okay, so what are the selective pressures on RNA viruses, and how to fight them? Well, let's talk just, there are a lot of them, like changing tissues when you go from bats to humans. There are changing hosts, like going to bats from humans, changing tissues, like when it spreads through your body. Or what we're going to talk about now is the very acute selection of antiviral treatment. And we know that a lot of times antiviral treatments select for drug resistance in this mathematical way that we like that people think about it. And how have we traditionally, what are the methods to combat that, to make antivirals more useful? Well, we know that combination therapy is nothing short of awesome. It's worked great for HIV and HCV. It's taken decades of research and a lot of funds. But combination therapy, of course, works if you have a frequency of 10⁻⁴, to have a single nucleotide escape mutation from one compound. You have two compounds, that becomes 10⁻⁸. You have three compounds, that becomes 10⁻⁴. We're reducing the probability that a multi-drug resistant virus is accumulated. A lot of people are really excited about choosing host targets. That is, viruses use a lot of proteins from us, including the receptors, including various membrane proteins for that proliferation. Maybe we can target the host targets because, we don't mutate that fast. And also, maybe the virus won't mutate away from its addiction to our host targets. That has not worked out as well in a lot of cases, but in some, it is a hopeful strategy. There's something called fitness cost. Maybe viruses are so perfect, their proteins are so perfect that we can target some active site, some surface, that any mutation in it that would escape inhibition by the antiviral would have a tremendous fitness cost associated. That is, it would be dead or sick. And that would be great if we could design fitness costs that way. I think an exceptional example of that is sofosbuvir, which is one of the drugs used to treat hepatitis C virus, in which it's really difficult to get drug-resistant variants, and they're pretty sick. This is hard to engineer. What I'm trying to do is figure out a way, and what we've been trying to do for a decade or so, is to try to figure out a new strategy, which is to choose drug targets so that the drug-susceptible cells, genomes in the same cell, make products that aren't loss of function, but instead of gain of function. And look, this poor fit virus, increased fitness virus, would then be surrounded by genomes that are making toxic products. And that's our goal. And I'm going to go through the progress, some of our thoughts and some of the progress we've made. Okay, so this is the idea. We're trying to have drug targets so that they're gain of function, deleterious proteins made by the other genomes, the drug susceptible genomes in the cell. Okay, so this is the organization. I'm going to talk about two paradigms we've worked on. One is, genetic dominance is frequently observed in oligomeric formation. For example, if this fit virus is making a capsid protein that doesn't bind to a drug that we give it, well, too bad, because its brothers, sisters, parents, et cetera, are making drug-bound capsids. And if they assemble into a chimera capsid, it's going to, on average, be drug susceptible. That was our original idea. And if that didn't work, I think nothing would. We'll talk about a new oligomer that we've started to work with and how we're trying to select inhibitors, how we're selecting inhibitors for it and its function. The second paradigm was a surprise to us. That is, within the polyproteins of positive-strand viruses, there are some cis-acting viral proteinases. And those, by the inability of, so these would have inhibited proteases, and the inability of this more fit proteinase to rescue cis-acting events leads to the accumulation of precursors. We'll talk about that in two cases, and that sort of complicated concept will be more clear. So these are the two paradigms that we found and extended a bit. Okay, so here's an example of a capsid. This is a cryo-EM structure from many years ago now of what happens during cell entry. So here's a icosahedral virion. Here are and around its five-fold axis are the receptor binding sites. What happens is this triggers, binding triggers endocytosis, and that acidic environment inside the endosome will cause the capsid to fall apart. It's important to remember that viruses are quasi-stable structures. That is, they have to be stable in the environment, but once they enter the cell, they have to fall apart and release their genomes. So there are two kind of drug targets here. One is the quasi-stable capsid. The other is the collapsed form. So we'll return to that in a little bit. Hocaprevir is a small molecule that is approved by the FDA for some purposes. It is a good inhibitor of poliovirus, all three serotypes, and is being worked on now for help in poliovirus eradication. We know that capsids, and what it does is it binds to these receptor binding sites around the five-fold axis. So that means 60 molecules per oligomeric capsids. We know that there are examples of genetic dominance in of molecules that inhibit capsids done in the lab of your own Eric Fried and also Adam Zlotnick at Indiana University for hepatitis B virus. So what happens to the endogenous inhibited proteins when in the presence of hocaprevir? Okay, so we're going to usual drug targets are the active sites of enzymes. So we're going to compare in poliovirus what happens when we have this capsid hyperstabilizer. Now it's a hyperstabilizer, not a dissociator. We're going to talk about an inhibitor of the active site of this NTPase that's needed in RNA replication. It's inhibited by a small molecule guanidine. And I'll refer to an inhibitor, rupintravir, that inhibits the active site of the proteinase of poliovirus because it behaves exactly like this. Okay, so our experiment is to select obtain drug-resistant variants. So hocaprevir-resistant variants, guanidine-resistant variants, rupintravir-resistant variants, and then test for whether in a mixed infection, those resistant variants are dominant or recessive with respect to drug susceptibility and co-infections. Now, people at this point say, don't you hate it at the end of a seminar when somebody says something that reveals they didn't understand, like your first 3 slides? So this question is something I get a lot. If drug resistance is going to be suppressed by some of your compounds, how do you get a drug-resistant virus in the first place? And remember, almost every variant exists at a frequency of about 3 × 10⁻⁴ in poliovirus populations. So if you dilute the virus enough and give every virus a chance to express its amazing qualities, then you can select for drug resistance. It's when it comes up in the quasi-species it's born in that we're trying to suppress its resistance. So we can select for drug-resistant variants through dilution. Okay, so what happens when you co-infect drug-susceptible and drug-resistant viruses? Here's what happens with guanidine, the enzyme that targets the active site of an enzyme. And here we're titrating into cells. We have a constant amount of drug-resistant virus to guanidine-resistant virus. And then we titrate in drug-susceptible virus and ask the drug-resistant virus, how do you feel about that? And the drug-resistant virus doesn't care at all. because the inhibited 2C molecules floating around in the cell are, pretty much monomeric or very low oligomeric species, and they just don't bother the drug-resistant virus. Their loss of function, the drug-bound ones. But here you go with bocaprevir, which is hyperstabilizing the virion. We have an MOI of 10 for each of these conditions, and as you titrate in drug-susceptible virus, you see transdominance of it inhibiting the drug-resistant one. So we're mimicking, of course, by this deliberate co-infection, what would happen inside a cell when you had drug-resistant virus coming up in the presence of drug-susceptible virus, at a ratio that would be much more than 10 to 1. Okay, our hypothesis, of course, is that in this case, let's see. So what happens in a mouse? What happens in a mouse, I actually did these experiments myself. So what happens in a mouse is that, so here are mice that express the poliovirus receptor infected by wild-type poliovirus. And so what happens is, after 4 days, here are untreated mice, and here are mice treated with guanidine. And what happens after 4 or 5 days, you start to see inhibition of the amount of virus there. And then you ask, well, what happens to drug resistance? Do you get selection? Here's the frequency of drug-resistant virus, and here's what happens when you, in the presence of guanidine, you see the expected selection for drug resistance at 4 and 5 days. You see these scales change. If you ask, is there drug-resistant virus in there? The answer is yes, at the expected frequency of 3 × 10⁻⁴, and you're obviously getting selection for it as the infection proceeds. Conversely, this is what happens when you have the capsid stable. The capsid stabilizer is you go 4, 5, 7 days into infection, and what you see is eventually, these aren't the best drugs in the world. What you see is the virus starts to be cleared, and you ask in the presence of the drug. And you ask, so what happens with drug resistance? And even at 7 days, there's no enrichment for drug resistance. Now, there could be a couple explanations for that. One could be a fitness cost, like maybe the drug resistant viruses that might be generated have low fitness. And the answer is they don't. In fact, they're more fit than the guanidine resistant ones. And then you might say, well, maybe they're not generated. But if you look at either of these populations and you say, are there drug-resistant viruses in there? The answer is yes, at a frequency of 3 × 10⁻⁴. So you are getting the quasi-species generating drug-resistant viruses. You've just blunted selection by choosing an oligomeric target. Are there chimeric viruses formed? Of course, this argument requires, these are supposed to be drug-resistant capsids. These are supposed to be drug-susceptible capsids. And there's the other case in which just there are homogeneous ones made from either genome. So to show this, of course, what you have to do is to pull out individual virions and ask, is this HA tagged drug-resistant virus in the same particle as the flag tagged drug-susceptible virus? And I think you guys can see the argument here, which is you pull down in a co-infected cell epitopes of both sorts. So yes, mixed capsids form. And we infer that that's the mechanism for the dominance of drug-susceptible capsid proteins. We did similar experiments with dengue virus. I'm not going to drag you through because they're very similar. We compared the core molecule of dengue virus and a molecule called ST148 that hyperstabilizes dimer formation, and an inhibitor, MK0608, that inhibits the polymerase. And we see very similar experiments, that is drug-resistant viruses are dominantly inhibited by drug-susceptible viruses, and drug resistance is suppressed in mice. So good. So, capsids work, and drug-bound capsid proteins can be dominant. Now, the question I sometimes get at this point is, but this is impractical. Are you planning on inhibiting viral infections by superinfection? And the answer is no. We co-infected to mimic the situation of the mixed infection that should arise within the cell as drug-resistant viruses are made. And I hope people get that and ask me more questions if you don't. But we mimicked by co-infection the situation here in the birth of a drug-resistant virus inside an organism or inside a cell. Okay, so that was our first paradigm, oligomeric proteins. What are we going to do now? Because I think we're pretty much out of drugs. Although, you know, this will be interesting to see whether it applies to HIV, and Eric and I have had a lot of conversations about this, or HBV, and Adam Zlotnick and I have had some conversations about that. But this is pretty cool. This is now a negative strand virus, and this is Lisa Seltzer, who actually came to my lab from Adam Zlotnick's lab, and she's an excellent biochemist. And she said, I want to work on influenza matrix protein, because there are a lot of inhibitors of influenza, as you know. There are terrible problems with drug resistance. And so she said, I want to understand the structure of the oligomeric matrix protein. Too bad we don't know too much about the molecular interaction. So I said, well, that's ambitious, but let's go. So what she did was, she's very scholarly. So she looked at the transition from neutral pH to low pH of what was in the literature. So here's a picture of a filamentous virion. You can see you can see hemagglutinin and matrix protein and neuraminidase on the outside. Underneath the viral membrane, you can see matrix protein assembled in a single layered oligomer. What happens when you treat a virion with low pH? It collapses into a different oligomer that is a multi-layered oligomer that looks like this in cells and in viruses. So what's going to happen when she purifies the viral protein and asks about its oligomeric state? What was known when she started was there are several structures of the amino terminal domain, which is highly alpha helical. And what she wanted to know was, what about the C-terminal domain, which had been implicated in oligomerization? So of course, she expressed the protein, full-length protein, which had been difficult because it, quote, aggregates, unquote. Well, as we say, one person's aggregate is another person's oligomer. So here we have the oligomer that she was able to make. And you see that it looks a lot like the low pH form, which makes sense because that's the low energy form that goes into the cell. Very irregular. We went to our colleagues and said, what about a cryo-EM structure? And they said, no way. Okay, but does this really look like the disrupted virion? Well, here is a cross-linking in mass spec of an intact virion at pH 7. And you see cross-links here between the C-terminal and the middle part of the N-terminal domain. But you see this extra one as though there's some new contact compared to the disrupted virion. And the oligomers she was able to make look like the disrupted virion. So that's cool. So our hypothesis is that she was able to oligomerize from wild-type virus was very similar to the multi-layered state in the disrupted virion. Okay, but Lisa, being the way she is, many mutations later to test these various interfaces that have been modeled and observed in the crystal structures, a lot of these mutations gave no assembly at all. Some of them continued to give this multi-layered structure, and one right here at this lateral interface gave rise to a single layered oligomer that we got very excited about, thinking it probably looked like the virion, the matrix structure in the virion. Okay, and it showed some of those crosslinks. In fact, some more of those crosslinks characteristic of the virion structure. So we felt like, okay, the N-terminus and C-terminus are at least disposed a little bit similarly with respect to each other. Let's go. So if you go to your cryo-EM friends with a structure like that, this happened with Hua Chu, who's at Stanford now, and in his lab, we said, well, is this regular enough now? And they said, yeah. And 6 months later, here goes. So this is the structure of full-length matrix protein from influenza A. You can see that what happens with the C-terminal domain is it kind of makes contacts with the N-terminal domain that are close enough for the crosslinks. In fact, given the amino acids, these are the red crosslinks that we saw that are characteristic of the virion and this mutant assemblage. Here's what the oligomer looks like. You can see that this N-terminus sort of drapes over onto the C-terminus of the next one. So there's domain swapping of various kinds and very good resolution even of the linker regions. So that's great. And what we're interested in is, as you might imagine, hyperstabilization of this and the low pH structure. Now, at this point, you show these oligomers and say you want to, look at, find small molecules to inhibit their function. And people say things like targeting protein-protein interactions is not a good idea because they're very difficult to disrupt with small molecules. To which we say, well, to ruin a party, you have to go. You have to go and stay a long time and be really obnoxious. And so what we're interested in doing is hyperstabilizing such structures. So how do you do that? So as I told you, these are melting curves of the pH 5.5 form of the purified matrix protein and the pH 7.5 form of the purified protein. You can see, and so this is binding of fluorescent hydrophobes. So what you see is increased fluorescence when the protein melts. Here's 65-degree melting temperature of the low pH form. Here's the much lower melting temperature of the pH 7.5 form. And what you can see, and then you screen in collaboration with a group at Stanford called ChemH, and they have access to various libraries and helped us screen these compounds. And so what you get are compounds, and we're asking for hyperstabilization of either the low pH or the high pH form. So what you see here is very cool. It's a combination of that whole screen. So here's the melting temperature at 5.5, and that's no change. And then as you go up by a couple degrees, what you see are compounds that hyperstabilize the low pH form. When you look on this axis, what you see are is no change in the melting temperature of the high pH form that's invariance, but now you see that there's some compounds that hyperstabilize that. We ran all these here, well, the ones that are light colors here, against lysozyme as well. And these are the ones that did not denature lysozyme or change its melting temperature in any way. So they seem to be specific, at least by that criterion, for these matrix protein structures. So the question is, what now? Do any of them, so as you might imagine, you know, we started synthesizing, having these molecules synthesized, and that takes a while. And here are some of our first results. So here's one of these compounds that hyperstabilizes the pH 5.5 form. Interestingly, a lot of these have peptide bond type structures in them. And you can see that after 12 hours of influenza infection, this is a multiplicity of infection titration. So we're going up in concentration of the virus. And what you see is a lot, oh, and the screen is live cells. This is a floor that identifies live cells. So no death, no death, no death by influenza until at 24 hours until you get to a higher MOI, and then you see that the cells start to die. And you see no difference after this first cycle of infection in the presence or absence of the molecule. If you let it go for 72 hours, that's now multiple cycles, you start to see in this range of multiplicity of infection spread through the plate. You're seeing an intermediate level of spread through the plate, and you do see an effect of this inhibitor. We infer temporarily that these pH 5.5 stabilizers don't inhibit cell entry in the first step, but they're inhibiting spread. But you know, there's a world of optimization of these molecules. These are our first results. What we're excited about is that we've been able, in an academic lab, to implement a high throughput screen to find hyperstabilizers of variants, and we're very interested in collaboration with anyone to help us try to realize this goal. Okay, so I think I told you about our first paradigm, which is we're excited about virally encoded oligomers. Capsid proteins prove to be what we call dominant drug targets, in which the drug susceptible subunits are dominant inhibitors. We're hoping that we can extend this to other kinds of viral oligomers. Now, the next paradigm I'm going to walk you through was, like I said, a surprise for us. So how do you make, when you inhibit a cis-acting proteinase, how do you make the drug susceptible genomes in the cell dominant inhibitors of this drug-resistant variant, that virus that will eventually occur? So our hypothesis here is that miscleaved precursors can be dominant inhibitors. And there's precedent for that in many viruses, starting with the alpha viruses a long time ago when Charlie Rice's lab worked on alpha viruses. And they showed that uncleaved precursors actually controlled the infectious cycle. And if you accumulated uncleaved precursors, you couldn't get anywhere past negative strand synthesis. And then, of course, Eric Fried has been working on small molecules that inhibit one of the cleavages in the HIV capsid that causes the inhibition of conformational change in that. So we start with the hypothesis that miscleaved precursors can be dominant inhibitors. Now we think about the polyprotein, and we think about the proteases and how they have to cut themselves out of the polyprotein. And some of those events occur only in cis. If that were to, and I'll show that to you, that were to happen, then no matter how good another virus was present in the cell, making a perfectly good protease, if this polyprotein isn't cleaving in cis, it will accumulate precursors. If those precursors are trans-acting inhibitors, drug-resistant virus will not grow out. So I'll tell you what we know about that. First of all, actually, these are the experiments that were done by Scott Crowder, and we published 16 years ago now, that got us on this whole line of experimentation. So this was where we found this surprise that I told you about, which is, so what we're doing now is we're transfecting in wild-type RNA. We're putting in mutant RNAs that have all these various mutations. And for each of those mutations, we ask the wild-type virus, does that bother you? And the answer is here. That is, I'm sorry, this is busy. All these red ones are mutations that bothered the wild-type virus. That is, this genome was transdominant, a transdominant inhibitor. And that's where we thought, okay, anything you do to the capsid protein pretty much is going to be a transdominant inhibitor of wild-type virus. 3C is one of the viral proteases that makes most of the cuts. It's the so-called major protease. If you talk to people in biotech and you say, what are you working on? They say, the major protease. And we say, of course you are. Too bad the genetic properties are not that good. Okay, so this is mutations in 3C have no effect on co-transfection with wild-type virus. So we assume that the drug-bound 3C enzyme just really doesn't bother, doesn't inhibit the drug-resistant one. However, mutations we made in 2A, that's a proteinase that makes only one cleavage in the polyprotein. It cleaves in cis itself, the bond between VP1 and 2A. So let me illustrate that here. These are all the cuts that are made by protease 3C. This is the one cut that's made by protease 2A between the capsid protein VP1 and this protease 2A. So, so the question was, and we found that dominant inhibition was true whether you inhibited the active site of 2A or this cleavage site itself. So you accumulate this precursor in two different ways in the co-transfected RNA virus, and you get dominant inhibition of wild-type virus. So there must be something horrible about this precursor. So because its accumulation was dominantly inhibited. You can make this precursor in rabbit reticulocyte extracts. It's kind of hard to detect in infected cells, but I'll tell you what we think is going on. Okay, so Jennifer Doherty, a grad student in my lab, who started a couple of years ago, actually wanted to study this specifically, having studied this paper and saying, well, so what do we know about why this precursor is dominant? And what if we had 2A inhibitors? So we don't have 2A inhibitors, but let's find out what's wrong with this precursor. And I had just always assumed, oh, this is just another capsid variant. This is just, we have, the VP1 has a ridiculous C terminus, and that's why it's a transdominant inhibitor. And she said, no, actually, there are two places it could be inhibitory. It could be inhibitory, remember, VP1 goes into the capsid, but 2A goes into the RNA replication complex. Maybe it inhibits at both steps. And without going through a lot of, so here we see that experiment repeated again with the 2As, with the defective 2A genome and with an irrelevant genome with another mutation in it that isn't transdominant. And you can see that over a time course of transfection, you see good inhibition, even at a pretty low mutant to wild type ratio. So you say, what's wrong here? What's wrong with the wild type virus that this other non-replicating RNA is bothering so much? And this is her slide about that. So does VP1 get incorporated into the capsid, or does VP1-2A get incorporated into the capsid, or does it get incorporated into the RNA replication complex? And without taking you through a lot of sucrose gradients, et cetera, the answer is both. That is, in the presence of this VP1-2A, what happens to the RNA replication complex is something terrible, which is RNA replication hardly occurs. To the extent you make variants, at about, a 20th, a 30th of the frequency, they're pretty unstable. They're more unstable than wild type variants. So we think that's what's wrong with precursors, and that's what's been inhibited. That's why it's really bad to accumulate them, and why we should target cis-acting proteases, because maybe they create two inhibitors, because they go to whatever complex VP1 would otherwise go to, and whatever complex 2A would otherwise go to, making two inhibitors. Okay, so let me tell you how paradigm 2 helped us solve a little puzzle in the inhibition of the dengue virus protease. So here is something from the literature, which is, a lot of people have been screening for inhibitors of the dengue virus protease. Here's the dengue virus genome. Again, it's a polyprotein. Many of the cuts are made by this protease, which is called NS2B3. So a lot of people have taken a small, a truncated protease domain and put it through high throughput screens for protease inhibitors. They find lots of protease inhibitors, but very few of them inhibit the virus. And, but there's one, which is called ARDP006, which is a pretty crummy inhibitor of the protease, but it's a 50-fold better inhibitor of the virion. Now, we're nowhere near to a drug here, of course, but we're trying to understand the genetics of drug inhibition and why some protease inhibitors work and not others. So there are many reasons to think that an inhibitor might be 50-fold better for the virion than a free protease, like maybe it concentrates in cells or something. But we, of course, had a hypothesis, that is, if this NS2B protease were making only cleavages in cis, then and those were toxic products to the other virions in the cell, then how many cleavages would you have to make? Maybe you could just make a few of those, inhibit a few of those cleavages, and you'd still make a toxic product, because this would be a gain of function instead of a loss of function in the targeted protein. Okay, so first question, does this protease cleave in cis? And the experiment here is to express this protein to be NS3,4A, in which there are these 3 cleavages made in a rabbit reticulocyte lysate. And what you see is this is a cleavage for a wild type, this wild type mini polyprotein. And this is what happens when you introduce in trans a good enzyme into an uncleavable polyprotein. And you see that it is not rescued by the protease added in trans. So each one of these cleavages is intramolecular and probably exclusively so. Okay, so what do we do now? So the hypothesis, as you might imagine, is these 3 cleavage sites are made in cis. Do any of these precursors, are they toxic? Well, actually, are any of these, is cleavage any of these sites inhibited by that molecule ARDP006? And if you ask about cleavage, and you make mutants in the other active sites, and you ask about cleavage of this junction, and yeah, cleavage is inhibited, but pretty poorly. This is disappearance of precursor, with about the same IC50 as inhibition of the protease with trans substrates. Ditto with this one. If you cleave both this one and this one, you get an IC50 of about 100 micromolar again. If you ask about that site right in the middle of it, NS3, what you see is an IC50 pretty similar to that of the inhibition of virus. And what you see is it also must change the conformation, because look, it can't make that cleavage either that got made more efficiently here. So there's something about cleavage there that has a very special effect on the conformation of this precursor. Okay, so the prediction is if this were the target of that inhibitor, because then our prediction would be that failure to cleave there would generate a toxic precursor. Is that true? The answer is yes. If you co-transfect, so this is how much virus wild type virus makes in a single cycle of infection. If you co-transfect it with a mutation in this site here, with a genome with a mutation in this site here, what you see is it has no effect at all. If you co-transfect it with actually equal amounts at this point of genome with a mutation in that cleavage site that is targeted by that drug, what you see is a pretty spectacular inhibition of growth in the single cycle of infection. And this is also true of a replicon, so we know that it's at the level of either translation or RNA replication, but we, for other experiments, show that it's RNA replication. Okay, so that means that cleavage in cis, the cis-acting proteinase, probably the reason that that drug is as effective as it is because it's generating a toxic product. So why not figure out what's the thing to inhibit to begin with when we screen for proteinases, inhibitors of proteinases that we want to inhibit viruses. So you can imagine that that's what we're doing going forward. So these are our strategies for dominant drug targeting that I just told you. We like this idea of cis-acting viral proteinases. What Molly Braun, a new postdoc in the lab, is doing is expressing the coronavirus PL-Pro protease and its surrounding cleavage sites to ask, are these occurring in cis? And are there any toxic products that could be generated by inhibiting those cleavages? So you can see where we're going forward with. We have these two paradigms. We want to identify small molecules, select drug-resistant genomes, ask for their drug susceptibility is dominant or drug resistance is dominant, and feel confident going forward that maybe we can learn to suppress drug resistance in the future in RNA viruses. Okay, so here's one of these people again asking these questions. Okay, you've shown that you can prevent the selection for drug-resistant viruses in the first round. What about when they spread? Well, that's a great question because here we are, we have this drug-resistant genome. Let's say it's inhibited 20-fold, 50-fold, 100-fold in this first round. What happens when it goes out and spreads to another tissue, you know, and I'd always thought that would be a problem. And so imagine my surprise when after 7 days in the mouse, there wasn't selection for drug resistance. But that kind of assumes the following method of viral spread, which is what we usually think about, that these viruses, a cell lyses, and these viruses go hither and yon, and they all start their own infection. Is that in fact the case? And how do viruses spread in tissues anyway? So, you know, there have been a lot of papers recently about so-called en bloc transmission of viruses, that maybe they mostly spread cell to cell, maybe they spread in little vesicles. And so we were, our contribution to that recently is an experiment that sort of failed. Okay, so here is a grad student, Jasmine Mashiri, in the lab, making colloids from human stem cells. And so you can grow these colloids and infect them with enterovirus 71. That's a picornavirus, very like, you know, a lot like poliovirus, but it's causing epidemics in Asia, mostly. And here are these green cells have been infected by enterovirus 71. This actin here shows that the polarity of these is apical out. So that's what an enterovirus would see, is the inside of the colon. Okay, so here we have some infected cells, and we're excited. We want to watch them spread, right? You know, what happens when multiple genomes spread together, et cetera, what happens in the presence of compounds. Too bad, because it turns out that what happens in this polarized monolayer is, you know, there is no spread of the virus within the organoid. Instead, this whole cell gets extruded. And you say, is it because it's apoptotic? And the answer is no, no markers of apoptosis. So this perfectly live cell is extruded from the monolayer, from the polarized epithelium toward the apical surface in a way that leaves it alive. And this is a process called extrusion. It's a normal way that polarized epithelia keep themselves, you know, fresh. But we didn't know that this happened with viruses. And you can forgive me if I drew in the quasi-species here. This is real data. This is the RNA, viral RNA within the cell. And here's our quasi-species, suggesting that in this case, anyway, viral genomes travel with their families. So if you have you can imagine, if you bring your horrible family with you, you may still never be able to, you know, to exert your special properties. Okay, so I just want to remind you that what we're trying to do is suppress the outgrowth of drug-resistant viruses and variants within the cell, and thus to prevent other events, but this is yet to be seen. And I want to say that, of course, this is why vaccines are so effective. If you think about this as a vaccine resistant, these hypothetical vaccine resistant viruses that we hear about, let's say it's resistant to a single monoclonal antibody. It's gonna make if it's resistant, it's still gonna make capsid proteins, spike proteins in the case of coronavirus that are from the vaccine susceptible genomes. And on average, you'll have drug susceptible proteins being made. So plus the vaccine response is polyvalent. So I really don't know too much literature about vaccine resistance. And I, for one, am not too concerned, and am pretty excited about the vaccine for coronavirus. Okay, so this is, these are the people who've contributed to this project in my lab. My lab is kind of small and young right now because these 4 people right here have now been hired by Gilead. And so, so this is my lab right now. This is Lisa, who did the oligomeric matrix protein work. This is Brian, whose graph you saw about the inhibition of the new influenza potential inhibitors. This is Jennifer, who wanted to do that old style, what's VP12A doing? This is Jasmine, who did the organoid work. And this is Dorothy, who's interested in bats and viruses. So these are our collaborators. I mentioned Wachu. The poliovirus work was done in collaboration with these people. I want to shout out to Yuncheng Park, who is my friendly program officer at the NIH and has been very supportive over the years. And I want to thank you for your attention.
Well, thank you very much, Karla. That was an extremely thought-provoking talk. I certainly really enjoyed it. One quick point I should make is to repeat the CME code, which is 31298. And so before we go to the chat box, I had one question. So with our maturation inhibitors, which stabilize an oligomeric complex, what we find when we select for resistance is destabilizing mutations that offset the stabilizing effect of the inhibitor. And those destabilizing mutants are actually addicted to or dependent on the inhibitors. Do you see that sort of thing with any of your, for example, polio or dengue mutants?
No, we haven't seen that yet. We don't have as good, so you're saying that maybe the pleconazole resistant mutants will be dependent on pleconazole?
For example, yes.
Yeah, we haven't seen that.
Okay.
In fact, they're perfectly fit. But that's good in your case. So if you were taking that compound, you would do cycles on and off.
Exactly. So it can't replicate without the compound being there.
Yeah.
Okay, so we have some questions. And by the way, to the audience, we have some time, so please feel free to send your questions in. Thanks for the talk. It's a really cool paradigm. Are there any examples of viruses that contain structural genes that would not be able to be targeted using your gain-of-function dominant inhibition strategy? Specifically, are there any viruses that are preferentially encapsulated by the capsid proteins that its own genome encodes, rather than being encapsulated by the pool of all capsid proteins produced by all the genomes in the cell?
That's a great question. And I don't know of any examples. Maybe someone in the audience does. The transencapsulation of other genomes is kind of a famous paradigm in virology. But that would be, if somebody asked how would the virus escape inhibition by drug susceptible subunits, that would probably be the way to do it. Somehow evolve a way in which the virus could cloak itself in membrane or something and thus keep its products to itself. incidentally, that's what we found for all the replication inhibitors of HCV. Even for like NS5A, which is an oligomer, and if you can inhibit it when expressed alone in trans, what happens during HCV infection is every little genome is tucked into its own little invagination. So you don't see mixing of the RNA replication proteins. And no one has tried, including us, to target the core protein of hepatitis C. But I think this hyperstabilization strategy, I don't see any reason why it wouldn't work in general. But you're right, if the genome kept its viral capsids to itself, it could escape.
So it's interesting to think about, protein-protein interfaces being poor targets for small molecules. But I think that argument doesn't hold so well for small molecules that are stabilizers, because they're not disrupting a large interface. They're actually promoting its increased stability. So I think for protein-protein interfaces and oligomers, I think it's a better strategy than trying to actually block the interaction.
Well, absolutely. And, for a long time, for the cell biologists out there, people couldn't figure out how Refeldin A worked because it blocks protein secretion, through these, multi-protein complexes. And it turns out that it does. It hyperstabilizes one of the complexes, so it can't go through the conformational change. They can't go through the conformational changes required for membrane fusion when you go from, Golgi to ER traffic. So there are plenty of examples out there of hyperstabilizing protein-protein interactions, and it's kind of a mystery to me why that's. I mean, maybe it is more frequently used, and I don't know about it, but I think it's just so much easier to do a whiz-bang screen by inhibiting an enzyme that's cleaving a fluorine trans or something. This is stuff of high throughput screens, and that's great, if you're dealing with blood clotting or something. But if you're dealing with something with an error rate like viruses, I think you need some different ideas.
And of course, with HIV, we already have drugs that target the enzymes, and in the long term, that might not be sufficient to control HIV. So we need other targets, and so we have to become a little bit more creative about the targets that we choose.
Right. And as you know, I'm kind of confused about HIV because there's the error-prone step is reverse transcription. And then once you have the DNA, you get into the genome, and then it, it has the error rate of DNA-dependent RNA transcription of the host, which I think, right, 10 to the minus 5, 10 to the minus 6. So the real error-prone step is reverse transcription. And then you're making pretty much the same RNA all the time from the integrated genome. That's if you only get one variant in the cell. I mean, but a lot of, you can get HIV people to argue all day about whether, infected cells are infected by one HIV particle or multiple ones. And that's going to differ in different tissues, but you're the expert on that, so I'm interested in what you think.
No, I mean, that is an ongoing debate in the field, and probably likely is determined by route of entry, that is cell-free infection, which is less efficient than cell-to-cell, which at least in culture results in the generation of multiple hits per cell. And when, of course, we know that recombination does occur with HIV, and that requires co-infection. So of course those events could be rare, and then could be selected for.
Well, I look forward to you all sorting that out.
Well, we'll try. Okay. Well, I'm not seeing anything else in the chat box, and it's 4 o'clock. So thank you very much. That was really a wonderful seminar, and we look forward to seeing more of your work coming out in the near future.
Thanks so much. Bye-bye.